Thank you very much for that introduction. This talk, I could see from the list of speakers in this series that a lot of the speakers are focusing specifically on technology, and I should warn you that this is sort of more of a basic science kind of talk. I think a lot of the work in my lab, which involves basic cognitive functional mapping, has important implications for technology and for future brain-machine interface applications, and for also has important neuro law and neuroethics implications. And I'd be happy to talk about that stuff in the questions, but the main body of this talk is going to be sort of more focused around data. So if you're not a data person, just go to sleep or surf on your phone for a while. So I'm interested in the brain. I'm a neuroscientist, and I'm interested in how the brain operates. There's a classical division of sort of theoretical approaches to the brain. You can study the implementation, which is how neurons actually compute. You can study the algorithm, which is how is data represented in the brain and how is it processed, or you can study computation, which is a sort of more vague kind of philosophical point of view about how the brain operates. And my lab mostly focuses on algorithm. Of course, if you're interested in the brain's algorithms, you have to acquire data at the implementation level, right? Because all of the data that comes in is going to be recordings from neurons that are either direct or indirect. And so we have to essentially aggregate data from usually some sort of indirect poor measurements, and then try to computationally model that data and infer something about the algorithm, which means either the representations or the processes that operate on those representations. Now, the brain is a multi-scale organ. For those of you who own a brain but don't ever look at it, this is a really quick overview of the organization of the brain. At the bottom, there's a bunch of neurons. And of course, in your brain, there's about 80 billion neurons, all wired together, each neuron connected to 5,000 or 10,000 other neurons. These neurons are organized into columns. So the cerebral cortex, which is that folded mantle on the outside of the brain, is actually a sheet that has 6 layers. And so you can look at the top of the sheet, like essentially like the top of a pizza, or you can look across the layers. And if you look at the top of the pizza, you'll see that the neurons are organized into columns. And at a broader level, these columns are organized into maps. And at a broader level, these maps are organized into areas. And then if you sort of take an entire brain, this is a macaque-monkey brain, a lateral view and a medial view of the macaque-monkey brain, and you, say, put an air hose in the brain and inflate it up into a beach ball, and then put some cuts in it and flatten it out. I'll show you how that's done in a few minutes. You can make a flat map of the entire brain, and you can draw the areas on the brain. And these are both anatomically and functionally defined areas. This is a monkey, so about 50% of its brain is visual, as shown in these various areas. And you can think of a functional area as a sort of a pepperoni on the top of a pizza. In your brain, the human brain, you have about 500 of these functional areas, and each one of these areas is performing a different sort of constellation of overlapping tasks. And all of these areas are wired together in a complicated network. And these 500 areas in the brain, in the cortex and the subcortical nuclei, each individual brain area is wired to about 50% of the other brain areas. And each one of these connections is a recurrent connection. So there's a feed-forward connection and a feedback connection. So that means that the brain is this really complicated, highly interconnected network where information is constantly flowing through the network and reverberating through these feedback loops at all different scales of the network. A system like this, if you have 500 parts, 500 isolated sort of modules, although, of course, every neuron can be considered a module, so there's really 80 billion modules. And if every single module is connected to another module through a feedback loop, what's going to happen is information is going to reverberate through this circuit, and it's going to create huge oscillations in the electrical activity in the brain. And a system like this, where information goes in and it percolates around and eventually flows out of the system at the other end, these systems are called nonlinear dynamical systems. And the problem with the nonlinear dynamical systems is we don't really have any math that describes these systems very well. And you all have suffered our ignorance. If you go into a building, any commercial office building, it has a thermostat, and it has a heating vent and an air conditioning vent. And the whole job of the thermostat is to measure the temperature and control the heating and air conditioning so that the temperature is constant in the building. And if you notice, most buildings are always, they're too hot or they're too cold or they're unstable. And this is because the feedback circuit is a nonlinear dynamical system that is difficult to control mathematically. And if you imagine scaling that up from one thermostat to 80 billion essentially units that all have to work simultaneously. This is a really complicated problem. And if we want to understand a non-neurodynamical system like the brain, we need to measure space and time simultaneously. We need to measure all of the individual units, which would be all of the neurons or all of the areas, and we need to measure their changes over time in their communications with other areas over time. And that means we need a measurement method that will measure in both space and time simultaneously. And the fundamental problem with human neuroscience is we don't have any device that can do that. So these are the questions that we ask in my lab about this system. Mostly we focus on the cortex, which is this overlying sheet that you can see here. We ask, how is this cortex divided into constituent functional areas and anatomical areas? What information is represented in each area? How are these representations mapped within each area? So you might have an area that represents, say, information about faces, but within that face representation, there might be some sort of substructure. For example, happy faces might be represented in one part and sad faces in another part. What aspects of these maps are common to the group, meaning all individuals, and what aspects reflect individual differences? How do these representations change over your life and over learning? And how are these areas arranged into task or state-specific networks? So one of the interesting things about the brain is, on a short time scale, it's just a collection of wires. You have 80 billion neurons. Each neuron is connected to somewhere between 1,000 and 10,000 other neurons. And all of these neurons are connected together with wires. And these wires, on a short time scale of, say, less than a few minutes, are fixed. The brain does not change. Because if you want to grow a new synapse, a new connection between one neuron and another neuron, it takes about 20 minutes to do that. So on a very short time scale, your brain is one fixed set of wires. Yet as you listen to me or you walk through your daily life, your thoughts are changing dynamically on a time scale much, much faster than 20 minutes. So somehow or other, this fixed set of wires gives rise to the emergent property of thought in a way that we don't really understand very well yet. But the nice thing about the fact that the brain's just a fixed set of wires is that there's a very close relationship between the wiring of the brain and the functional activity in the brain. Because obviously, if two brain areas are hooked together with a wire, then there's probably going to be some functional relationship between those brain areas. So in principle, we can try to understand the brain by either looking at the wires, which is the physical connectome of the brain, or we can try to understand it by looking at functional activity. And these two things, in the end, should give us the same answer. They should give us isomorphic results. So I'm interested particularly in functional mapping of the brain. That's the idea that different portions of the brain are specialized for different functional activities or functional representations. Now, the idea that the brain has functional specialization is not new. It goes back actually a couple hundred years. The first thought that maybe the brain was functionally specialized occurred when people realized that people with bullet wounds that they recovered from showed different kinds of bullet wounds to the brain. They showed different kinds of deficits. And this idea that the brain was functionally specialized was sort of adopted by a now discredited field that you've probably heard of called phrenology. The phrenologists had the idea that you should be able to functionally parcellate the brain by looking at the bumps that overlie the brain, the bumps on the skull, and that if there was a large bump, that probably meant that brain area was large, and that meant that the person who possessed that large bump would have a certain functional capacity that was specialized for whatever underlie that bump. And the phrenologists analyzed a lot of people with a lot of bumps, and they came up with complicated maps like this that tried to assign functional areas to individual locations in the brain. And this was a complete and total waste of time. In fact, this is the poster child for bad science. But the phrenologists, although completely wrong about the facts, they were in some sense right about the basic idea that there is functional specialization in the brain. The problem is they didn't have any method for actually figuring out what the brain function was in any area. And that's what we have today that they didn't have. So say we want to measure the function in the brain. What options do we have? Well, this is a map that shows all the different ways we can measure the brain. This is time here. This is different time scales for these different measurements. This is different spatial scales for these different measurements. And all of the measurements down here at the bottom that are very small spatial scales and that expand over all time scales, all of these methods are invasive methods that you can use in animals with no problem, but in humans you can only use them under medical necessity, and most of them you can't actually use in humans at all. Nobody patch clamps humans, for example. All of these methods up here are suitable for humans, and they all have limitations. First of all, they're all, if you notice, none of them can measure individual neurons. They're all bulk tissue measurements, and they move over different scales. But there's a worse problem with them than that, and this slide summarizes it. These are the 4 standard methods we can use with humans today. Electroencephalography measures electrical activity in the skull. Magnetoencephalography measures magnetic activity. FNIRS measures blood flow in the skull using optical methods. FNIRS is exactly the same technology that the doctor uses when they pull a pulse oximeter on your finger, and it shows when your pulse is going because it changes the blood, changes from red to blue when the arterial blood is replaced with venous blood. And in FNIRS, we just put a big laser on your head, and we measure the change in your head as the blood changes from red to blue. And then we have MRI, which is a sort of a magical method. It's bulk chemistry. It measures changes in oxygen concentration in the bloodstream, which is related indirectly to neural activity. And now these 2 methods are portable. These 2 methods are not portable. These cost millions of dollars. These 2 methods measure time, but they don't measure space. And the reason is the skull acts like a big sort of smoothing filter, and it smooths out all the fine spatial detail. And so if you measure electrical activity in the brain using EEG, you can tell when something happened, but you can't tell where it happened. And because there are 500 different brain areas, which brain area was giving rise to a measured activity on the surface of the skull is obviously important. These methods measure changes in oxygen in the blood supply that are much slower than neural changes, and so these methods don't measure dynamics. In fact, there's no data in MRI at a time scale faster than, say, 2 seconds. So this is a problem. We can measure time or we can measure space. We can't measure space-time. And whoever measures space-time first will be a rich person. So I'd like to show you the kind of data we measure in MRI. I tend to use MRI in my lab because I want to recover maps. I want to recover functional maps across the surface of cortex, and I'm willing to give up the dynamics. So we have a brain. The brain is inconveniently folded up inside the head, so we inflate the brain, as you saw, then we put some slices in it and we flatten it out. If we did this to your brain, we'd have something about the size of a large pizza. This is the left hemisphere. This is the right hemisphere. This is the visual system in the middle. This is the prefrontal cortex at the left and right. This is the motor and somatosensory strip here and here. And this is the auditory system here and here, just to orient you. Now, on the surface of this brain, we're painting brain activity. This is metabolic brain activity as measured in MRI. These are changes in blood oxygen. Neurons act as little chemical engines, and when a neuron fires, it extracts sugar and oxygen from the bloodstream. And those changes in oxygen use, we can measure using MRI. Now, if we record MRI while someone is watching a movie, say they're fixating on a small spot in the middle of this movie, and they're just watching this movie, you can see that the patterns of brain activity change dynamically over time. Now, these changes in blood flow are much slower than changes in neural firing. A neuron can fire 100 times a second. We only see changes in blood flow at a time scale of a second or 2. There's really no information below that time scale. So we're an order of magnitude slower than the neural signals, yet MRI is indirectly related to sort of the integral of the neural signals over time. And you can see that the patterns of brain activity change over time. Red here means a lot of metabolic activity, probably a lot of neurons firing. Blue means very little brain activity, probably few neurons firing. And where the brain activity is high changes quite a bit as people watch this movie. And the reason for this is anything that you can think about in this movie must be represented in the brain somewhere, right? We're not dualists. We think the brain is all you have. So if you can see something in the movie or think about something in the movie, it must be in the brain. So all the little edges of these objects are in the brain. The fact that these are 2 people kissing is in the brain. If you think, oh, I like kissing, and you think you have a positive affect about kissing, then that would also be represented in the brain. If you think, oh, I remember when I got married, that was a great day, that's an association that you dredge up from memory, that will be represented in the brain. Everything that you can think about is represented in this brain activity somehow or other. And the job of functional mapping is to find systematic relationships between the stuff that happened in this movie or the stuff, the latent variables in your head that are associated with this movie that we can't measure directly, and brain activity. You can think of this as a big, gigantic regression problem. There's a bunch of stuff in this movie. There's 100,000 points we measure in the brain. We just want to find, for every single point we measure in the brain, all the stuff in the movie that it cares about. Okay? And of course, if you build a perfect model of the brain, that means for any point in this brain, you will be able to perfectly predict what that voxel does. A voxel is a volumetric pixel, a small point in the brain that we measure. You'll be able to predict exactly what that voxel will do under any state of the world that could ever occur. Right? Okay, so I kind of made it clear MRI is not a perfect method. I use MRI, I've used it for years. I hate it, and I hate it because it's not a perfect method and it's got limitations. First of all, it's indirect. It doesn't actually measure neural activity. It measures changes in the blood vessels nearby neurons. And that means there's an indirect nonlinear coupling between the thing we care about, which is neurons, and the thing we're measuring, which is blood vessels. So we're not really measuring from the brain. We're measuring from this thing, which is actually a plasticized version of the blood supply of the brain. That's really what you should think about we're measuring here. Because we're measuring blood vessels, and these blood vessels are essentially receiving indirect coupling from millions of neurons, each one of these small volumetric pixels is receiving input from about 1 million, or summing input over 1 million to 5 million neurons, then the spatial resolution of our MRI signals is low. Neurons are organized on a column level. We cannot see those individual columns. We're going to see something on the scale of something that's called a hypercolumn, which is integrates over large information. And these signals are very slow. All right. So now for the next couple minutes, I'd like to turn to another issue, which is in the face of this overwhelming complexity, these voxels could do anything. In fact, they do everything. And we need to try to figure out what the brain is doing. How should we solve the problem? I'd just like to spend a few minutes on philosophy issues, because the methods my lab uses to solve these problems are very different from other labs. We use the same machine, an MRI machine, but we approach the data very differently. So the goal of science is to measure your system, then create an explanation of the system whose dimensionality is lower than the measurements. Otherwise, you've just made your problem worse. And then to be able to predict. We want to predict as much of the variance in our system as possible, and we want it to generalize to novel situations, and we want to generalize over individuals, right? And this is essentially, the government actually pays us for this thing here, these predictions. They don't actually really care about explanations. Only scientists care about explanations. Engineers don't care about explanations. They generally just want to predict. So in engineering now, a lot of you have heard of deep networks. Deep networks basically just jump right over explanation. They go from measurement to prediction directly, and trying to figure out why a deep network is doing what it's doing is virtually impossible. This dimensionality is actually larger than the measurements. So in science, our goal is to build an explanation, but it's a certain kind of explanation. It's an explanation that predicts. Now in MRI, the history of MRI comes from the history of cognitive psychology, and in particular, a certain flavor of cognitive psychology called additive or subtractive factors logic, which relies on very heavy linearity assumptions. Generally, an MRI experiment, a typical MRI experiment, will have two conditions like faces versus places. I show faces, I show places, I record from a voxel, I have the time course of the face versus the place, and I can subtract the response to faces versus the response to places, or this is objects, excuse me, not places, over time, and I can find, say, a small spot that I will call a region of interest, which is a face-selective area. And this is fine as far as it goes, but remember, if you had a brain area that, say, responded to bowling balls, and bowling balls were not in your image set, you would not be able to determine that this brain area actually cared about bowling balls, because if you only have a contrast here, you can only talk about faces versus places. So these methods don't tend to generalize very well outside of the exact experimental circumstances that were used to fit the model. This is a common failing in our field, in psychology. On top of that, psychology follows a very strict point-null hypothesis testing framework, where you propose a hypothesis, you design an experiment that optimally discriminates between that hypothesis being true and random data. You design that experiment, you run that experiment, and then you can discriminate whether or not your hypothesis is likely, whether the data are more consistent with your hypothesis or with random data. This approach, which is very common in psychology and biology, has really fallen on hard times in the last 10 years. There have been there's a lot of discussion about replication failures in psychology, and especially in the more difficult and complicated and abstract areas of psychology, like social psychology. Of course, social psychology is much more difficult than cognitive psychology. Cognitive psychology only involves one person. Social psychology involves two people. Therefore, it has to be more complicated, right? For those of you in psychology who are depressed about the replication crisis, I want to tell you, you are by far not the worst people. The replication crisis in biology is a log unit worse than in psychology. It's a horror show. Remember, every psychology student has taken a few statistics classes. Until recently, statistics classes were not required in any biology program. And therefore, biologists have generally had real bad problems with replication. And a lot of molecular biology results have failures to replicate. And it's fairly typical if you're working in a pharmacological company that you're going to make a drug based on some molecular biology finding. The very first thing you do is you have to replicate that finding because you pretty much can't trust the published literature. So this is an ongoing problem, not just in psychology, but in all areas of multi-organism studies where we have to aggregate data across a lot of noisy measurements and variable individuals to try to reach a conclusion. It's a really bad problem. There's another thing that I personally just want to point out. This is a personal issue that comes from the fact that in my lab, we usually do small n studies, meaning we'll collect data from 1 people or 2 people or 4 people, rather than a typical psychology experiment where we collect data from 40 people. And the reason for that is just my history. I used to be a neurophysiologist. In neurophysiology, you'll get one animal, you'll get all your data, and then you'll get a second animal just to make sure the first animal didn't cheat, and then you're done. You only need 2 animals. Psychology, no one would do that. That would be insane. You get 50 animals, and you average them all together, because after all, most psychology experiments are undergraduates. The undergraduates were up late the night before. They're not that reliable responders, and your data is noisy. Okay, if you are of the opinion that you should collect large numbers of subjects, you are essentially implicitly assuming that the data are like this. Essentially, if we take all the variance in an individual subject, you're saying that a bunch of the variance is individual variance and only a small amount of the variance is group variance. After all, if the opposite was true, if most of the variance was group variance and only a small amount of the variance was individual variance, you wouldn't need 50 subjects. You could just take one subject, and that would be a good model for everybody. If you assert that you need 50 subjects, that means you're saying that the situation is over here, that the individuals are highly variable. That may be true, but remember what happens if you build a group model in this situation. That group model will only account for a small fraction of the variance in any individual person. So it won't predict any individual person very well at all. It'll only predict the group average. And therefore, you're not going to be able to use that model for, say, diagnosis or for classification in the clinical setting or for predicting the individual behavior of any individual because the individual variability is so high. So my personal opinion is that the argument that we need to collect large numbers of subjects in psychology studies is not a logically valid argument. Either you only need a few subjects, and you collect a lot of data from those few subjects, or what you're doing isn't all that interesting because you're predicting an amount of variance in the group that is so small as a proportion of the total variance, who really cares? Okay. So I have an approach from my lab that comes from physiology and a branch of engineering called system identification that overcomes sort of all of these problems in my opinion, and I'm happy to argue about it, because I spend half my time arguing about these things. Let's take a situation like this. We start with a very complicated data set, like, say, movies or stories, and we collect some brain activity data. Now, obviously, anything in these movies or stories must be represented somewhere in the brain, but we don't know what, and we don't know where. So we're going to take this data, and instead of proposing a hypothesis beforehand, we're going to propose a hypothesis afterwards, after we collect the data, and we're going to propose a bunch of hypotheses. So, for example, we might think that there's a part of the brain that extracts the motion energy from the movies. We might expect that there might be a part of the brain that would extract the contours from the movies, or say the surface normals that would tell us the orientation of the surfaces, or maybe the semantic content of the movies, like what objects are present in the movies. And we can take the movies and we can move them into all of these different feature spaces. And now for every single one of these 100,000 points in the brain we measure, we can fit a model that optimally maps those new features into the brain activity. And in MRI data, for those of you who do MRI, typically you have enough data to fit about, say, 10 or 20,000 features, 10,000 features, and you can divide those features up into these feature spaces however you want. So now we have a bunch of models. We have 100,000 models for each of these 4, in this case, hypotheses. Now we collect a new data set where we show in some new data and we collect some new brain activity. And now we do not fit with this data. This is the fit set. This is the test set, or the estimation set and the validation set. In this case, we take these movies, we extract the features from them, we multiply them times the weights we fit in this stage, and then we get predictions. And now for every single voxel, the voxel will tell us what feature spaces it cares about. If this voxel cares about motion energy, then this prediction will be really, really high, and this prediction for semantics will be low. If the voxel is in a semantic area, this prediction will be high, and this prediction will be low. So now every voxel tells us what it cares about. And because we've got separate fit and test data sets, the problem with p-hacking and replication largely goes away because the noise in this data set is independent of the noise in this data set. And not only can we compute statistical significance, which is necessary but not sufficient for doing science, we can actually get predictions. We can tell you how much of the variance, the potentially explainable variance in these signals, are we explaining with our model. And that means we can tell when we're done. If the potential of the totally potentially explainable variance in my experiment is, 100%, say it's 80% of the total variance, and my model predicts 80% of the variance, then I know I'm done. I can't build a better model than that. Okay, so this approach provides a clear method to test several competing models, makes all assumptions about the models explicit, permits use of complicated natural stimuli, which means you can generalize to natural stimuli. This is a complicated arcane thing I won't get to. Provides functional maps in each individual subject. We do this at the individual subject level. And we get prediction accuracy and generalization. So let me show, so we did a lot of movie experiments for several years, and I won't describe the experiments because they're trivial. We had people watch movies. We recorded brain activity. That's all there was in the experiment. And then we fit these kinds of models, and I'll just talk about 2 models. This motion energy model, which describes low-level statistical structure, the edges and the movement of edges through a field. And we think there are parts of the brain that care about motion energy. And a high-level model that was essentially just labels of the objects in the movies. Like, was there a dog? Was there a person? Was there a cat? And now we can fit both of those models simultaneously to the voxels in the brain. This is the visual system at the back of the head. And you can see that the blue model, which is motion energy, fits really well in these low-level visual areas. And this high-level semantic model fits really well in these high-level visual areas. So that's kind of what you would expect based on what we know about the organization of the visual system, which starts off coding really simple features of objects and then progresses hierarchically through several stages of processing until you have representations of high-level abstract concepts. Now you can look at the data more carefully if you wish, using methods I won't go into in detail. This is called a semantic tree. This is actually a tree that comes from WordNet. WordNet is a hierarchy. This was built by linguists over about 20 years, sitting in a small room with large quantities of coffee, and they said, a cat. What is a cat? Okay, a cat is a feline. A feline is a mammal. A mammal is an animal. An animal is an organism. An organism is a thing. Right? And they built this giant tree of an inheritance hierarchy of all the nouns in the English language and many verbs. Verbs have their own trees. And you can basically extract, in this case, 2,000 of these nouns and verbs, the 2,000 most common nouns and verbs in the English language, and you can build a model for each one. And so all of these, and this is just the tree, you can't really see it here. I'll just tell you that all of these yellow things here are animals. All of these green things are people, different kinds of people like nurses, doctors, lawyers, firemen, so on. These purple things down here are vehicles. And now you can, for these different segments of the tree, you can look to see where these attributes are represented on the cortical map. So for example, all of these red areas are coding for places, as I recall. All of the green things are people. Anything you see green is people, and so on. So you can find these pretty rich maps, and you always find 2 things when you make these maps. The first is that any given concept, like the concept of people, is represented in multiple places in the brain. And every single place in the brain, like this little spot right here, will represent a constellation of related semantic concepts about people. Now the visual stuff pretty much stops right about here. This is the best map because it implies that this stuff is signal. It's mostly noise. Most of the visual signal is stopping at the anterior border of the occipital lobe. Now you can use these models to decode information. And to do this, you simply run the data backwards through the model. And so if you show somebody a movie and you have a model of an early visual area like primary visual cortex and how that represents edges, you can extract, you can basically use the model to recover the edge map from the movie data. So here's a movie we showed somebody in the magnet. Here's the decoded movie. And this is just an edge map of this and an edge map of this, so that you can see that the edges actually correspond pretty well here. This is a decoder built from one visual area, primary visual cortex, area 17, and, or area V1 in your brain, it's called. And you can imagine if you have 500 of these areas, you can decode a lot of information, right? Because here we're only decoding one brain area. This is a decoder that decodes information from a different brain area. This is a few high-level brain areas that represent the semantic content of the scene. So you can see that here there's a, okay, so now we're at fish. You can see it gets fish, body of water, water, swim. Okay, there's no sky, got that wrong. This is not a whale, it's a manatee, but we don't have manatee in our set, so it thinks it's a whale. That's a pretty good mistake. Here's where the thing kind of breaks down because there was no snowcat or snow in our training set, so it gets sky, but it thinks the snowcat is a building, and it thinks this is the body of water. But that's an honest mistake, right? This decoder actually works remarkably well, and again, it's only decoding from a few high-level brain areas. If we were decoding from 500 brain areas, all 500 brain areas, whatever information is encoded in there, we could build a great decoder that would, basically reify everything that is sort of emergent and present in current conscious thought. And I'd like to show you another example of this quickly from stories. This is another pretty simple experiment. We put people in the MRI machine, and we had them listen to stories. And these are stories from the Moth Radio Hour. Some of you may have heard. It's from Public Radio International. Moth Radio Hour is sort of stand-up storytelling. People get up in front of an audience, and they tell usually personal autobiographical stories about their lives. These are awesome stories because they're semi-professional, at least experienced storytellers, and they're telling the most compelling story they can because they want the audience to vote for their story. They're competing against other people. So this is a great experiment because it's very easy. You go, you lie in the magnet, you close your eyes, you listen to the stories. This is the only MRI experiment I've ever done where we don't have to pay our subjects to have them perform the task well because they're happy to just go in there and listen to the stories for free. They listen to about 3 hours of these stories over several sessions, and then we aggregate the data together and we model it. And we're going to use several models. We have the brain data, we have the stories. We take the stories, we break them up into several feature spaces, a spectral feature space, which is the short time sound spectrogram. That's the representation of your cochlea. A phonemic representation, syntax, and semantics. Now, for each of these feature spaces and for every one of the 100,000 locations in the brain, we fit a separate model. So every one of these 100,000 locations gets 4 separate models. And then we take a new set of stories, we extract their features, we multiply them times the models we got in this stage, and then we calculate predictions. So every voxel will tell us which of these feature spaces it likes. Now there's a problem with language, which is language happens often on a very fast time scale. This is 1,000 hertz, is very, very fast. This is a millisecond, and changes in your vocal cords and your glottal pulses occur at the scale of milliseconds. Phonemes occur at a very short time scale. And our MRI machine has a Nyquist frequency about here. We can't really recover any information from the MRI machine at a time scale faster than about 2 seconds because we are not measuring neurons, we're measuring changes in blood flow. So all of this data down here is very difficult for us to pull out. What we can pull out of is changes in narrative, the semantics, and emotion. We can pull out a little bit of information about phonemes, but the signal-to-noise will be very low. That's just a limitation of the method. One of the problems you have in these sorts of naturalistic experiments is that all of the stimuli are correlated with one another. So the sound spectrogram is going to be correlated with phonemes, that's going to be correlated with syllables, that's going to be correlated with words, that's going to be correlated with syntax. So all these models are collinear, so you have to do some sort of variance partitioning analysis. Fortunately, psychologists have a very good way to do this. This is an old method in psychology from 50 years ago, really. And now you can use modeling and variance partitioning to map across the surface of the brain 3 different, in this case, feature spaces: the spectral feature space, the articulatory feature space, which is essentially phonemes, and a semantic model. And you can see that most of the models fit really well in some part of auditory cortex, which is this circle here on the left and the right. The only model that fits well outside of auditory cortex is the semantic model. The semantic model fits everywhere. What is semantics? This is a lexical semantics model. It's a model of the meaning of language. So as you're listening to language, of course, you're processing phonemes, you're processing the sound spectrogram. You don't care about any of that. What you care about is the meaning of the story. And this map of meaning turns out to be distributed everywhere across the brain. So we can actually look at that map. And that is shown here. Now, on the top are 1,000 semantic concepts. These are the 1,000 most common nouns and verbs in the English language: cats, dogs, doctors, fire trucks, whatever. And if we take all of the brain data and aggregate it together, we can recover a semantic space whose structure really isn't all that important for the purpose of this talk. And then we can aggregate together different parts of this space just to show you what they look like. So for example, all the emotional words are down here in this space. They're kind of purple. All the social words are red. These visual words are in green. And we can paint across the surface of the cortex the semantic selectivity of each of these 100,000 voxels. So for example, the voxels in this purple patch here, which is on the medial wall on the precuneus and the retrospinal cortex, just above that, all of these voxels are tuned for mental concepts like thinking, dreaming, wishing, believing. Whenever those semantic topics appear in the stories, these voxels become activated. All of these voxels, these red voxels right in the middle of the temporal-parietal junction, these voxels tend to be responsive to social concepts like mother, father, daughter, marriage, divorce, death, things like that. Social concepts, social things that happen to people, and social identifiers of people. But if you notice, all of the colors are broadly distributed in these really rich, complicated maps. There isn't just one red spot. There are red spots all over the place. There are purple spots all over the place. There are green spots all over the place. And that's because each of these sort of semantic domains, like visual, tactile kinds of things like rough, smooth, bumpy, green, social concepts, numbers, all of these different kind of domains of semantic information all appear in different distributed, interdigitated networks distributed across the surface of cortex. If you want to play with this data, we actually have an online viewer for this. You can go to my website, and there's a whole bunch of brain viewers. This is just one of the brain viewers. And you can interact with this data. So here I am playing with the brain viewer on the... Sorry, you can't see it very well because of these lights. But we're clicking around red voxels here in the brain. And you can see that each time I click on a red voxel, it gives me a statistical significance. It tells me how accurate our model is and how much of the variance we can predict. And it gives me a word cloud that is the 20 or so words which, when they appear in stories, we predict would most activate this voxel. And you can see that every time I click on a red patch, it's essentially the same kinds of words: father, mother, wife, husband, refused, married, murdered, pregnant, things that happen to people and their families. Now I'm clicking on a green patch. This is in the posterior parietal cortex, and you can see that this is a number-related patch. This is 1, 2, 3, 4, 5, days, weeks, months, money, and time. All of those things tend to be represented in this number network, and this number network, again, has many, many nodes in it. Here's a number patch, here's a number patch just forward of the temporal parietal junction, and here's a different number patch down here on the ventral aspect of the temporal lobe. So all of these different semantic concepts are represented in these rich networks across various brain areas. Now this works really well. We decoded vision, and you saw that the vision decoder was actually good. And now I'm going to show you a failed experiment. This is our language decoder. So now we're trying to decode the stories at the left here based on this semantic model and this brain activity that you see here and here. And now this is the story, and this is the word cloud. Oh, sorry, that's supposed to repeat. This is the word cloud. And you can see that it gets the gist right sometimes, but I would never publish this. This is really just a horrible abomination of a model. It does not do well at all. And the reason has to do with the time scale. If you see a dog in a movie, that dog's probably going to be around for a while. It might march back and forth across the screen until there's a screen cut or the dog goes out of frame. It's going to be there. If there's a room, the room's going to be there for a while. Things in movies tend to happen over a fairly long time scale. Things in language don't happen over such a long time scale. They happen very, very quickly. If I tell you a story about, Joe came home and the dog jumped him in his lap, and then his wife came in and yelled at him, this topic of the story has changed 3 times in just a few sentences. So all of that data happens very, very quickly, and we just can't recover that very well from MRI. So you might ask, well, these are people who are listening to stories. What happens when they read stories instead of listening to stories? We got at that by just showing people the same exact stories by reading and by listening. These are the predictions of the semantic model for reading and listening. This is reading, predicting, reading, listening, predicting, listening. And this is the cross predictions. This is reading, predicting, listening, predicting, reading. You can see that the cross predictions are very good. That might suggest that this semantic information is immune to whether it comes in through reading or listening. And that would be true. These are the two semantic maps. This is a semantic map for listening. This is the semantic map for reading. You can see that these maps are almost indistinguishable from one another. You can't tell the difference between the representation of the meaning of a story, whether it came in through listening or reading. So this isn't really, these maps are not maps of spoken speech. They're maps of the meaning of language that are apart from whether it was spoken or read. Another thing you might ask, since I talked about movies and I talked about stories, is wait, we had a semantic map for movies, we had a semantic map for stories, how do these things relate? And that's an interesting question that we pursued recently. Here's the semantic maps for listening. So all of these semantic areas were identified, or semantic patches, I haven't really shown you the areas in any statistical sense here, were identified from people listening to stories. And this is the same brain, but a completely different data set. These are the semantic areas that are sort of identified, or the semantic tuning of the brain while people are watching the movie. Is there a relationship between this? If you see a movie and it has a dog in it, and you hear a story about a dog, what is the relationship between those semantic representations? So to get at that, we noticed that at the anterior border of the occipital lobe, remember, is the posterior, most posterior part of your brain. That's where your visual system lives. At the anterior border of the occipital lobe, there are a bunch of voxels that are tuned for both language and movies. So they will respond in a language experiment, and they will respond in a movie experiment. Voxels behind this border will only respond in movies, and they won't respond when you're listening to stories. And voxels in front of this border will only respond when you're listening to stories. They won't respond to movies. So you can statistically go in and identify a little strip where blue is voxels that only respond to stories, green is voxels that respond to movies only, and red is voxels that respond to both. And you can see that there is a close connection between the responses to movies and responses to stories. They're right next to each other. The question is, what is the semantic tuning of the movie voxels and the story voxels? And I won't show you, in the interest of time, how we get at that. Instead, I'll just show you the result. This is the semantic tuning, using this color map down here, of the voxels behind this strip, the voxels that are only tuned for vision in movies, and the voxels in front of this strip, the ones that are only tuned for stories. And you can see that in almost all cases, there's a systematic relationship between the tuning behind the border and the tuning in front of the border. The voxels tend to be tuned for the same things. If you have a voxel that's tuned for in this visual information behind the border, it will be tuned for that'll be visual information in movies. It will be tuned for visual information in stories immediately anterior to the border. And we think this is actually, and there's probably anatomical connections from the visual part of the brain into the language-related part of the brain, so that when you're watching a movie and you're thinking about its meaning, you're generating internal speech, that those parts of the semantic representation are connected. There's one place where this fails miserably, and it's this purple patch here. You notice this purple patch does not have any kind of analog in the visual area. This purple patch only appears when people are listening to stories, and there's nothing in the visual system that is related to this purple patch. This purple patch turns out to be semantic concepts in stories related to mental functions, like thinking, dreaming, believing, wishing, faith, things like that. And those don't really have an analog in the movies because we didn't code for any sort of abstract concepts. So they're just not represented in the visual system at all. Now, you can aggregate the data together to come up with a single semantic map for both movies and stories that shows that this meaning information is broadly distributed across huge swaths of the brain. And you can use this, if you wish, to sort of decode audiovisual movies and to reconstruct both the visual content of the movies and the soundtrack of the movies, although decoding the soundtrack will be a lot worse because MRI is very slow. There's one, in the last 3 or 4 minutes, I just want to mention one complication in all of this. I've giving you a story that sounds a lot like modern age phrenology. There's a bunch of brain areas, and they're all tuned for functional things, and each brain area has one specific set of similar functions, things that it does or things that it represents. And, you know, okay, well, there might not be one representation of dog. There might be 20 representations of dogs. But still, there are visual and general semantic areas that reflect, you know, the meaning of dogs. We suspect there's multiple areas that represent, say, dog and every other concept because they actually reflect different attributes associated with those concepts. So, for example, a dog-related patch near the visual system might contain information or represent information about how a dog looks. A dog-related patch near the olfactory bulb might represent information about how a dog smells. A dog patch near the auditory system might represent information about how a dog sounds. And then you're going to have abstract patches that are tuned for dogs that represent sort of your episodic information about dogs, like, oh, I remember when a dog bit me when I was a little kid, for example. All that information is going to be represented in this distributed network, broadly distributed across the brain. But everything I told you makes it sound like these networks are static. And the problem with this neuroimaging data, and in fact all psychology and neurophysiology data, is these representations are not static. And we know from neurophysiology this is true. If you record from a prefrontal cortex neuron in a monkey doing a working memory task, say, and then you record from that same neuron while the animal does a different task that's maybe not a working memory task, maybe it's doing a mathematical reasoning task where it has to count the number of dots on the screen, the neurons in prefrontal cortex will change the way they operate. And in the prefrontal cortex neurophysiology world, this is called mixed selectivity. We have an analog of this in vision. If you record from a neuron in an intermediate visual area, like V4, and you have an animal look for a dog in a movie, and then you switch, you'll find a certain tuning curve for stuff in the movie, right? Depending on what that neuron is tuned for. And then if you have the animal look for, say, a car in a movie, the neuron's tuning will actually shift toward the car. And you can see this on the single cell level throughout the entire brain. The magnitude of these shifts depends on where you are in the brain, but all neurons tend to shift their tuning curves to a greater or lesser extent. So we did an MRI experiment to see if we could see the same thing in humans, and we did the simplest experiment we could possibly think of. We had people watch movies, and we just had two conditions. First, in one condition, attend to humans. In another condition, attend to vehicles. All you have to do is watch for when there's a human, hit the button. Watch when there's a vehicle, hit the button. Simple task. Easy to do, and now we basically just fit two models. We have a category model that reflects how each of these individual voxels, these 100,000 voxels, are tuned for about 1,000 categories when the person is searching for people and when they're searching for vehicles. And now we can compare the tuning in this 1,000 category space under these two conditions to see if the tuning of the voxel changes when they're searching for one thing or another. So this is that same semantic tree you saw earlier. I know it's hard to believe. This is the same thing. But this is actually a tree you just can't. I wish these lights were not on. You cannot really see the branches of this tree. But basically, the root is about here. And these are a bunch of people. These are all the people-related concepts here. These are all the vehicle-related concepts here. And the size of the circles here indicates the amount by which the tuning for that voxel for all voxels over the whole brain increases when the person is searching for that concept. So when they're searching for people, the responses of all of the voxels in the brain to people goes up, and when they're searching for vehicles, the responses of all the voxels in the brain to vehicles goes up. So it's as if the voxels are changing their filtering properties based on what you're trying to do. And this is another way to look at this data. Here we have a semantic space. It doesn't matter what these axes are. They're just the principal components of the semantic space. Here's where humans should be in the semantic space. Here's where vehicles are. And when this is the distribution of individual voxels, they're shown in red and blue here. And this is the state, the tuning of the individual voxels in the semantic space when you're searching for humans. And you can see most of the voxels are crowded over here towards humans. And now when you're searching for vehicles, this is exactly the same semantic space. This is the humans template, this is the vehicles template. And you can see that the voxels now kind of shift up here. And now they're tuned more for the vehicle-y part of the space than for the human part of the space. Right? Except for these blue voxels actually shift away from the attended target. And now you can look at where these voxels are on the surface of the cortex. That's shown over here on the right. You can see that most of the voxels are either gray, they don't shift their tuning, or they're red. And some subset of the voxels are blue. So the red voxels are places in the brain where the tuning always shifts toward the attended category. And the blue voxels are voxels where the tuning shifts away from the attended category. And this is a little inside baseball, but since this is Missouri, I assume you guys all know about the default network, which is the part of the brain that becomes active when you lie in the MRI machine and tell people not to pay attention to anything and to just turn their brain off. And of course, what people do when you tell them to do this is they start talking to themselves, mostly saying, God, this MRI experiment is really boring and uncomfortable. When do I get out? When do I get my money? I'm hungry. I want lunch. And in fact, the default network is very highly overlapping with the internal speech rumination network. They're almost identical. So these areas, these blue areas, prefrontal cortex, lateral prefrontal, the TPJ, and the precuneus turn out to be part of the default network. So our theory is that if I put you in a task like search for people, you're sitting there in the task, okay, I gotta find people, I gotta find people, I gotta find people, I gotta find people. You do not want to respond to the part of your brain that is generating these images and potential targets of people, because that would not solve the task. The task is to respond to the external world. So you try to essentially damp down or detune the parts of your brain that are generating the internal signals and shift the parts of your brain that are paying attention to the external world so that they're more tuned to pick up these signals that you're searching for. And that mechanism is called a matched filter. And you can see that this matched filter mechanism actually changes these maps quite a bit. Here's the map in the neutral state. Here's the map when you're searching for humans, and it turns out in this task, humans are mostly green in this color code, and this is the task when you're searching for vehicles, and vehicles are mostly purple, and you can see the proportion of green and purple changes. So the tuning all over the whole brain changes when your task changes. And the amount that the tuning changes is directly proportional to how far the brain area is from the sensory system. So the visual voxels actually change the least. The voxels that change the most are the ones up in the front of your brain, the prefrontal cortex. And this is a really weird and important fact. Well, let me give you one more intuition about this. Imagine that you have some semantic space, okay? And this is just different semantic concepts where their distance in this space is their similarity, right? Body parts and talking are more similar than body parts and vehicles, right? And I want to represent this space by the activity of a bunch of filters that are tuned for little parts of this local pieces of this space. So I have a bunch of basis functions that are tiling this space. Now, what happens is when I tell you to pay attention to humans, the voxels kind of shift so that they're representing this part of the space with more fidelity. And that means that you'll be able to discriminate humans from non-humans really, really well, but other stuff in other parts of the space you won't be able to discriminate as well as you could because you don't have as many filters there. Then when I tell you to pay attention to humans, you shift up and pay attention to humans, and the other part of the space is spared. This is mathematically equivalent to stretching this membrane about the space. Now, this match filter mechanism is very powerful because it means you can take a small amount of fixed hardware and reallocate it for the task at hand. But it really screws up everything that we think we know about the brain. Because one of the fundamental assumptions, working assumptions that we all make in neuroscience is that a neuron acts as what is known as a labeled line. Meaning when a neuron fires, it always means the same thing. If I have a neuron in a part of the visual system that's responsive to faces, whenever that neuron fires, I know that there was a face, right? And that's important because that's how the downstream neuron knows what the upstream neuron is telling it by virtue of the fact that that upstream neuron is always telling you about faces. But in this case, all the neurons in the brain are changing their tuning based on the task. And that means that the downstream neuron that's receiving information from the upstream neuron can't determine what that information means unless that downstream neuron knows what the task is. And that means that all the neurons in the brain have to know what the task is. Well, where is the task-related information? Nobody has a clue. We have no idea how these tasks are instantiated or where they're represented or how that information is transmitted. There's this problem that everybody's talking, but we don't know but they're talking different languages at different times, and we don't know where the signals are that determine, that allow the system to know what language is being spoken at any moment. This is a really fundamental problem for neuroscience, and I have no clue how to deal with it. All right, so I told you that semantic information is broadly distributed in these networks for vision and for language. These networks are very, very rich. They obviously reflect long-term knowledge that you have about the world. And this knowledge does not reside in one location for one concept. It's broadly distributed in these very rich interacting networks. There's a systematic spatial relationship between the functional subnetworks for semantics in vision and the functional subnetworks for semantics in language. And this probably means that these networks are related to each other and that they're closely anatomically coupled. And these functional maps, however, are plastic and change on task, and we don't really know what that means or how it works. And we're going to pursue that in the future once somebody has some vague idea of what to do, because right now it's just a big open hole of a problem. Anyway, thanks very much for your time. Sorry I went over a little bit. Somebody should have yelled at me. I was supposed to be talking to the mic, and instead I went and stood over there the entire time. I will repeat your question, Jeff.
So you showed maps of meaning and other neat things. For some maps, like the visual area was pretty contained, and other maps were highly diffuse. And I'm wondering if you have any neat stories about things that are preserved across individuals and things that vary wildly between individuals.
So one of the nice things about this voxel-wise modeling framework is that you can quantify how much of the variance in your data set is reflected in individuals versus the groups. And in these MRI, these naturalistic MRI experiments, generally, oops, that was the wrong slide, generally about a third of the variance is group variance, and about 2/3 of the variance is individual variance. And the way you can do that is you can take all of your MRI data and do principal components on it, and you find out usually for an MRI experiment you need something on the order of 100 principal components to account for all the data. And the first, say, 5 to 7 principal components will account for 30% of the variance, and those will be common across all the subjects. And then all the other principal components will be individual differences. So this is a problem for psychology. This loops back to what we were talking about at the beginning of, do you want to build a group model or an individual model? My personal philosophy, and probably one that you will share based on talks I've seen you give in the past, is that you should build both, because some of the variance is shared in the group and some of the variance is individual. And after all, we want to predict everything. We want to predict everything. That's the megalomaniacal goal of science. So we should build 2-level models, a group model and an individual model. So in this case, we have 4 individual people. And you can see, now you can ask yourself, are these maps the same or are they different? This is from a language experiment. And most people, when I take a poll in the audience, they say, well, no, they're different. And that's because 2/3 of the variance is individual variance. But 1/3 of the variance is common here, right? You can see everybody has these 2 big red spots in their temporoparietal junction, right? Everybody has a big red spot here in the precuneus. Everybody has maybe a green stripe and a yellow stripe and a green stripe. Green stripe, yellow, green stripe, right? But it's, it's not, if you squint and hope, it looks like maybe there's something common here. Now, usually in MRI, the way you deal with these data is frankly not good. You average all the brains together, because every brain is different, just like every individual person's ear is different. How you average them together is a hard problem. In fact, it's not a solvable problem. So you use some criteria that kind of enable you to morph 2 brains together. And what that does is it loses most of the information. It essentially acts as a smoothing function. It throws away a lot of the detail. So we have another way to do this, which I will not go through the math, because trust me, you don't want to know. All you have to think about is this at a vague level. Imagine that the flattened brain is like a pizza, and the functional areas are like pepperonis on a pizza. Then if you assume that everybody has the same pepperonis, they're just slightly morphed because of the different shapes of brains, you can write an algorithm that will treat these different functional areas like balls and springs and morph them all together and basically find the right solution. And you can build a brain atlas that way. So this is the brain atlas for language. You get this out of one experiment. This is one experiment with maybe 5 subjects listening to 3 hours of data each. And we're getting 160 semantically selective brain areas. The color here tells you about the semantic selectivity of the brain area. And the thing to remember is this is a group level model, so every single subject has all of these areas. And this is a model that allows us to map from any individual into this model. So this model is essentially a model that relates the individual anatomy to the functional data. So if you give me new anatomy, I can predict where the functional areas are. So we have ways to do this, but this is still, I think, an open area of statistical modeling because most people in MRI they just don't want to deal with individual differences at all. They just only focus on the group model. And if you're only focusing on the group model, then smoothing the data is not going to cost you anything because you're just smoothing out the individual differences anyway. Yes?
As soon as I got oriented to this map, the first thing I looked for was Wernicke's area. But Wernicke's area is nothing special, is it?
Wernicke's area is just another, semantically, it's like around here somewhere, right? It's going to be like somewhere, some near the temporal parietal junction areas. We did not, and Broca's area, here BA is here. BA is also semantically selective, especially the top part of it. So Broca's and Wernicke's both have semantic signals in them. That doesn't mean that they're only doing semantics, or even maybe mostly doing semantics, because you'd think if you're looking at Broca's and Wernicke's, you're looking at something that really has to do with speech production, and this task is only speech comprehension.
Wernicke's should be active for speech comprehension also.
It is active.
Okay. What could you have
And it's tuned. Well, I don't have a Wernicke's localizer, so I'm not sure exactly where it is. It's going to be somewhere in here, right?
What could you have predicted of this from lesion data?
Nothing. So this is this is a good question. So the gold standard for brain mapping is neuropsychology, right? And in neuropsychology, generally, you want to find a double dissociation. You want to find a lesion in a subject where they can't do one task, but they can do another task. And you find a different lesion in a second subject where they can do the first task, but they can't do a second task. And now you have a double dissociation. You can say, okay, these areas seem to be bottlenecks and critically important for these functions. Most of these semantic areas are not bottleneck areas. They're just part of this broadly distributed semantic network. And if you end up with a brain lesion, you'll just kind of suck a little compared to how you did before. And in fact, that's true for most brain lesions that you get in prefrontal cortex or these parietal areas. If you end up with a small brain lesion, not a massive one, you just get kind of worse at a lot of things. And I think that's what's happening here. So if you had a lesion in Wernicke's, that information is represented in a bunch of other places. So you just won't be as good as you were at tasks, but these systems have a lot of redundant information in them.
Thanks.
And just so you know, we are doing language. So all the MRI data, except as far as I know, maybe 2 MRI studies on language, is all about comprehension. All of it. Why? Because if you speak, you have to move your jaw. And if you move your jaw, first of all, your head moves, which is the worst thing that can happen in MRI. And second of all, you change the magnetic susceptibility. Having moving body inside the magnetic field changes all the magnetic fields, and it just causes all kinds of signal to noise problems. So we have a bunch of experiments on language production with using specialized head stabilizers that my lab manufactures that keep you from moving your head. And then some of them are doing explicit speech with your mouth, which is bad because your mouth is moving. So we also have typing, which is better because your fingers are moving very far from your head. And then we also have this experiment on internal singing. So we tried doing some pilots on internal speech. We did internal speech. That was very difficult because people were very inconsistent. Then we did internal poetry. That was a little better. But then we realized if you want to make sure that people will do the exact same thing every time so that you can get multiple repeats of your data, which you need, have them sing. So they learn to sing a song while a metronome is playing, and so they know the beat, and then they can sing the song, and they're doing it completely internally. So now we have internal speech of a known piece of text while the metronome is playing, and that's pretty cool data. And we are actually, the student who's doing this, Sarah Popham, she actually has 2 conditions. She has the regular song and the parody song from Weird Al Yankovic. So you sing both of them. So now we have the exact same song tempo and the exact same song prosody, but different semantic meaning in these 2 different cases, and we can compare them against each other. It's a fun experiment. So we're working on that. Maybe next year we'll have that. Well, thank you very much for your time. It was very fun. Thanks.